Okay? So, good morning everyone. I am quite excited for today's session kay hopefully mas ma-better understand ninyo ang ato ang topic, which is all about a short talk on how to catch up with midterms, enhancement on basic quality control, carbohydrates, lipids, and question and answer should you have any clarifications or if meron ba kayong gusto i-discuss outside of the topics that I have prepared today and kung ano ba yung direction natin for finals. So catching up on midterms. So according here that the hardest subjects for you is carbohydrates, followed by lipids and quality control statistics. Nganong naglisod mo class sa carbs and lipids? Is it because of the bulk of the discussion, tama? Aside from the bulk of the discussion, and siguro minor lang ang correlation with the lecture, I think one of the factors din is nakalimutan na siya. Ano siya nalimutan? Kaya I remember class, 'di ba, ang first quiz na to that was before intrams pa. And for finals, we could still have this enhancement discussion. Okay? Especially since you have your comprehensive examinations. So, moving on, let's have our enhancement discussion on basic quality control statistics, of which, according to the survey, these are the topics that you find the hardest. First is yung Westgard multi-rule system and yung errors. So this is a bit not necessarily surprising kasi ang Westgard multi-rule system class, I guess, meron talaga tayong mga nakakalimutan, ma-mix up, especially in terms of to interpret or kailan ba siya na-discover basing on the performance of your quiz. Then for the errors naman class, there is actually not much for me to discuss in terms of the errors kasi somewhat it's raw memorization, or if not raw memorization, then I guess context clues and correlation. Paano ba identify ang random and systematic error? Paano ba i-identify ang shift versus trend? And to understand that shift and trends are all systematic errors. So without further ado, let's try to look into first the quality control Westgard multi-rule system, of which I will be answering majority of the practice na charts. Okay? So for the interests of time and para mataas-taas yung cover natin, I'll be answering 5 charts. Sige. So first, let's look at the chart 1. So how many errors exist? How many errors ba daw class ang present on this Levi Jennings? So obviously, you have the first error and ibilang pa natin class. Meron pa bang other errors dito? How many consecutive days nga dapat class between shifts and trends? 'Di ba for shifts and trends, it has a consecutive error of 6 or tama ba ko? 6 or 7 for shift and then 7 days for trend.
Yes, sir.
Okay. So, we have here 1, 2, 3, 4, 5, 6, 7. And actually, class, this is a downtrend. Bakit siya downtrend? Kasi if you look at it, there are 7 consecutive days going downward. Pero class, medyo tricky siya nga, no? Because by definition, typically kasi ang trend is only at one side of the mean. Okay? At typically, ang trends talaga class, it is one side of the mean. Pero ito siya class is a type of trend that passes through the mean. And actually, meron yun siyang specific label according to the Westgard multi-rule system. Pero para dili kita ma-overwhelm, let's just simply label this one as a trend. And you know, class, that trends are systematic errors. So you would identify this error as a systematic error, specifically a trend. Now, 'di ba, according to definition, trends should be for 7 consecutive days. So if ang question class is on what day nangyari ang error, you would count the 7th day. So 1, 2, 3, 4, 5, 6, 7. So dito na araw nag-occur ang error. Okay? Now, how about the next na error? Ito. This is one value outside of 3 standard deviations. So, this is 1, 3, S. And class, is this a random or systematic error?
Random po.
Okay, this is a random error. I think it's quite easy for you to distinguish systematic and random error just by looking at the Levi-Jennings chart. Ganito kasi siya, class. For most instances, yung mga systematic error natin is usually consecutive siya. For example, what are errors that are consecutive? 2 to S, you have your 2 to S, 2 to S. 'Di ba consecutive ang 2 to S? Let's say for example, dito and dito. So you have two values outside of two two values outside of the second standard deviation. So that is consecutive, so it is a systematic error. Another systematic error, you also have your 4, 1S. So let's say, for example, 1, 2, 3, 4. So these are four values sitting just outside the one standard deviation. So this is 4, 1S. And remember, class, na ang 4, 1S is 4 consecutive days. So you would label it as a systematic error. Now, another point of confusion na usually gina-clarify sa ACUA is naabay 4-1S wherein the values are outside of 2SD or the value is outside 3SD. And the answer, class, is yes, because 2SD and 3SD, all of them are outside of 1SD. Meaning to say, class, if makikita mo og ing ani nga pattern, all of these values are sitting outside of one standard deviation. Two of this value is outside of two standard deviations. But all of the four values are outside of the first standard deviation. Meaning to say, class, this is actually a 1 4S na rule. Ay, 4 1S rather, I'm sorry. This is a 4 1S rule na nasira. Okay? Klaro ba, class?
Yes, sir.
Okay. So, in just this Levi-Jennings, balik muna tayo sa Levi-Jennings alone, we could identify two errors. The first is a trend, and the second is a 1-3S. Where is rejection? Ang rejection natin, class, we will base our rejection dito lang sa Westgard multi-rule. Okay? So, according to the Westgard multi-rule, di ba, class, ang for rejection is everything other than 1 to S. So, for day 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, for day 11, class, kayo would be rejecting the run. Why? Because this is a 1, 3S error. A random error that is subject for rejection. Okay? So now let's look at the Levi-Jennings chart number 2. So, you could already identify, class, 2 to S error. What else? Meron pa bang error na na-miss? I think wala naman. Yeah, wala na. So this is a 2 to S error. And as I've mentioned, if the error occurred consecutively, then this is a systematic error. Now, on what day did the error occur? Class, ha? 2 to S yung error. On what day did the error occur? So on this case, class, if 2 to S ang tinanong kung anong day nangyari siya, you would say that on the second day, doon nangyari ang 2 to S na error. Kasi on the first day, hindi mo pa man alam kung ano ang result ng second day. Okay? Kasi if ito lang yung tingnan natin, class, this is a 1 to S error. If 1 to S siya, is it subject for rejection? Hindi. Kasi 1 to S siya, it is a warning rule. Nalaman mo lang na 2 to S siya the following day. So on what error did the 2 to S error occur? The day after the first warning rule. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. 16 nangyari ang 1 to S. 17 nangyari ang 2 to S. Okay. On which day are you going to reject the run? Is it on day 16? Is it on day 17? Or is it on day 16 and 17? Again, class, you're going to look at the errors chronologically. If you're going to look at day 16, it is a 1 to S error, which is a random error subject for warning. Hindi mo pa alam kung ano yung result for day 2. So of course, you're not going to reject the run on day 16. Pero by day 17, you found another 1 to S error. And dalawang consecutive na 1 to S class is already a 2 to S error, which is a systematic type of error subject for rejection. Meaning to say, you're going to reject the second day of the run, day 17. Hindi day 16 and day 17. Only day 17. Kasi hindi ka naman maka-go back in time yesterday para i-reject ang run yesterday. So only reject the day kung kailan nangyari ang systematic rejection rule, which is 2 to S. I hope that is clear. Now, let's look at another chart. Levi Jennings number 3. I think ito yung pinaka-obvious. One value outside of 2 negative SD, another value outside of 2 positive SD. And this is what? 4 standard deviations away. So is this R4S or is this 41S? Obviously, both of them has 4 in it. Pero iba ang meaning ng 4 nila. For R4S, ang meaning ng 4 dito class is ang distance. Whereas for 41S, ang number of consecutive errors. Meaning to say class, R4S is something that is, ito class, itong R4S, this is a random error. So ito yung instances class wherein there is consecutive errors in the run but it is not a systematic error. Kasi class, di ba, ang tendency kasi ng systematic error is usually at one side of the mean lang talaga siya. Usually. Dito class, sa random error is on 2 separate occasions, the distance is positive 2 and negative 2 standard deviations away. And that is an indication of a random error. Kasi randomly nag-shift agad-agad. Okay? Okay. So, let's try Levi Jennings number 4. This is a 4-1S error. Bakit? Kasi 1, 2, 3, 4. Are there rules? Ganito siya class ha. Ganito siya. So, you have 4 days involved. 1, 2, 3, 4, 5. Day 6, day 7, day 8, and day 9. The question is, how many errors are subject for rejection? Meron bang error subject for rejection class?
Day 9, sir.
Day 9. Day 9 is the error subject for rejection. Meaning to say, day 6, day 7, and day 8 are the control runs that you are going to accept. Bakit siya accept? Kasi in the first place, the values are less than 2 SD. Within acceptable range sa class, less than 2 SD. Ibang usapan naman yun class if ganito yung run. Kasi this is already a 2 to S error. Okay? For Levi Jennings number 5 class, do you see an error here? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So this is an example of a 10x bar error. By the way, class, just to clarify, if the runs are only one side of the mean, even if 12 days sila, di ba class 12 days ito, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. 12 days all and all. You will label this error as a 10x, not 12x. Any value greater than 10 on one side of the mean, 10x ang tawag niya na error. And as I've mentioned, if the errors are consecutive, or if the days of the run are consecutive na nagkaroon siya ng error, then this is labeled as a systematic error. Okay? Systematic error ito siya class. Now, the question is, di ba, ito yung day 10. What if day 9 pa lang class? Are these runs subject for rejection? Yes or no?
No, sir.
Hindi. Pero what if class, meron tayong 2 to S na error dito? Let's say for example, ganito yun. Now, identify kung ano ba yung errors na present. You would actually identify a 1 to S error on this day alone. Pero again, 1 to S is not a subject for rejection. Okay? So merong mga instances class na in just one type of error, in just the 10 days or 12 days, merong other errors na present. Pero dito sa example ko class, na merong 1 to S inside of the 10x bar rule, 1 to S is another error, but just not subject for rejection. Okay? Sige, bigyan ko pa kayo ng other example regarding that kasi medyo complex naman to siya class. So let's say class na on day, bilang ha, day 10, 11, 12. Sige, ito yung days class. Let's say for example that on day 13 and 14 kay merong 2 to S na error. And then let's say for example na wait lang ha. Okay. So ito yung nangyari class. You have 10 values. You have 10 values outside. 10 values on one side of the mean rather. Now, etong runs na ito class, you actually can identify a lot of errors. Okay? So, bilangin natin class. Sige, ano ba yung first error na ma-identify nyo dapat class?
2 to S.
2 to S? Final? Final class?
1 to S, sir, tapos nag 2 to S.
Yes, you would first identify 1 to S. On the next day, you would identify 2 to S. What is the next rule? What is the next error that you're going to identify? Ang next na error na ma-identify class is, ano kaya?
10x bar, sir.
10x bar. Kasi 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Dito na-identify yung 10x bar. After 10x bar, meron pang isang error na ma-identify. And that is
For 1S.
for 1S. So how many errors are all in all? There are 4 errors or in all. Now, what if ang tanong is how many errors are subject for rejection? Remember, meron kang 1 to S, 2 to S, 10x bar, and 4 1S. Now, there are 4 errors. Tama, 4 yung errors. Now, let's change the question. How many errors are subject for rejection? And you know that only 1 to S is a warning rule. Meaning to say, there are 3 errors subject for rejection. Ganun lang siya, class. And then, i-identify mo on which days are you going to reject the run. So, kailan ang last day ng 2S, kailan ang last day ng 10x bar, kailan ang last day ng 41S. Pero class ha, di ba I've told you na any value greater than 10 on one side of the mean is still 10x bar. Pero kano sa ka mag-start reject og run sa 10x bar? Sa first day na nitungtong siya og 10. Klaro ba, class? So,
Yes.
kano sa ka mag-reject og run for 10x bar dito? Kano sa ka mag-reject for the 41S error on the last day of the 41S? Okay? Ideally kasi class, when you're going to make the Levi-Jennings chart, for every error kasi class, dapat kasi i-troubleshoot mo siya para iriran mo ang control until ma-within normal siya. So ideally, there should not be multiple errors in one Levi-Jennings chart. Again, for every run na dapat i-reject, i-troubleshoot nyo dapat yung machine so that the control would be within control limits. Ideally. Kaya dapat class, in normal conditions, there are no multiple errors in one Levi-Jennings chart. In normal conditions, in typical conditions, dapat walang multiple errors ang LJ chart. Okay? So naklaro ba ang LJ5 class? Mas na-gets ba?
Yes, sir.
Yes, sir.
Yes, sir.
Alright, sige. Now, for the rest of the slides, class, gi-include ko kasi yung 20 na practice na ginawa ko. Meron ba kayong gusto i-solve in particular? Or na-answer nyo na? So, I am assuming na wala na kayong in particular na gusto i-ask regarding the 20 charts that I've sent as a practice. So, kayo na lang ang magsagot ng class, ha? Now, let's move on to the second topic for basic quality control statistics, which is all about errors. Sige. For errors class, there is nothing for me that I can add na something that you don't have to memorize or study. But what I did do na lang class is, do you study with AI? Nag-use pa kayo ng AI to study class?
Yes, sir.
So, actually, I would really suggest that you're going to study AI. Pero, do not use AI as a reference. So, what you should do is add a reference for you to be able to synthesize what you have learned. In this case, yun yung ginawa ko dito, class. I am not ashamed to admit that I, as an instructor, am using AI. Kasi, class, AI is in fact a tool. So, as long as dili lang ta magsalig sa AI, then we have no problem. And please do study smart because AI class, it really helps you in terms of studying. I'm sure the majority of you are using AI. So the same, ito yung ginawa ko for this part of this discussion. So what I did na lang class is with the help of AI, I was able to synthesize what are the key differences in random and systematic error. And ito yung mga gusto ko i-emphasize on the discussion. Ang random error class affects precision, whereas ang systematic error affects accuracy. Kasi in the first place class, diba, if you're going to look at a random error, typically, ito yung nangyayari sa random error. Ito typically ang nangyayari sa random error. Diba class, for the 7 consecutive days, merong isang day na randomly nag-akyat siya. So, that affects repeatability. For example, ito, pipetting variation, bubbles, and operator variability. Do you think, class, na ang bubbles ng tube nyo, if i-run ko again ang test, do you think that we have the same number of bubbles? Do you think that the bubbles in my tube and the bubbles in your tube has the same volume? Hindi. Meaning to say, class, yung error nyo is something that I could not replicate. Meaning to say, there is a problem in the repeatability. There is a problem in precision. Another, pipetting variation. Do you think, class, ang mali nyo na pipetting is magawa ko din siya, mapipet ko din siya in the same way na wrong ang pagkapipet nyo? Hindi. So again, it affects repeatability. It affects precision. And that is random error. For systematic error naman, class, again, it affects accuracy. Ang pagkatotoo sa actual na value. Okay? So ano ba yung examples? It could be calibration, it could be reagent lot change, or it could be instrument bias. Ang key thing that you have to remember for systematic error class is typically, it lies on one side of the mean. Typically. Pero that is different if merong trends and shift. Okay? Of which trend and shift is a type of systematic error. Other than trend and shift, you also have your Westgard multi-rule, which includes 22S, 41S, and 10X bar. And if you've noticed, class, itong mga Westgard multi-rule natin for systematic error are those days wherein the runs are in consecutives. Okay? 2 days, 4 days, or 10 days, or greater than 10 days. Okay? Now, this is another table for differentiating random and systematic error. Okay? So for this part class, I'm just gonna leave this for your reading. And now, you also have your shift versus trend, both of which are systematic errors. So this is quite easy to remember. Kasi shift, sudden. Trend, gradual. madali lang din siya i-identify class because typically among the examples sa inyong trend, if nakita ninyo sa PowerPoint, ang mga examples sa trend kaya naka-emphasize ang gradual increase, gradual deterioration, mga ing ato na ay typically gradual or basta ginaingon niya na hinay, then that is already a trend. Basta mabagal ang change, that is a trend. Now, on which days ba sila nangyayari? Greater than 6 days ang shift, greater than 7 days ang trend. Okay? Then, ang shift is one side of the mean, continuous upward or downward movement. Yun yung sabi ko, class, that typically, a systematic error is lying outside just one part of the mean. Typically. And that definition, class, also agrees with the definition of shift. Pero ang trend, class, just like sa example ko kanina, a trend can actually traverse the mean line. Okay? Pero class, by the way, ang trends natin, it is not necessarily a subject for rejection. Kasi possible kasi class na ganito ang mangyari. So let's say that this is the mean. And let's say that this is your first standard deviation. Now, what if magpakita ako sa iyo ng trend, class, na pwede siya i-accept? So this, class, is actually a value that is acceptable. This has a downward trend. Acceptable siya, bakit? Because all of these values are lying on one side of the mean. While there is an effect in accuracy, pero still the values are within control. Yun yung mga instances, class, wherein trends or even shift is acceptable. Okay? So here is another table on how to differentiate shift and trends. Basta sudden, class, shift talaga yan siya. Okay? Basta may slowly or gradual, then that is a trend. Okay? So now let's move on to carbohydrates. And according to the survey, this is the topics that you find the hardest. The enzymatic as well as the chemical glucose assays. So ito siya, class. It's not surprising na mahirapan kayo kasi I guess understanding the discussion is easy. Memorizing the discussion is quite difficult. So unsa ang kailangan nato ipang memorize for enzymatic and chemical glucose methodologies? If not memorize, then maybe at least familiarize. Remember, class, that ever since we've moved on from midterms, 'di ba, I requested for you to have a shift in how you study. The first is yung request ko that you should study in algorithms. Other than algorithms, you should also be able to study through tabulations. Why? Because, class, if you were the ones that made your own algorithm and tabulation, it is actually quite easy for you to understand, or if not understand, then at least have a spatial understanding on the discussion. Kasi pasabot ako sa spatial, class. Spatial. Kasi if you're going to practice making algorithms and tabulations, class, you're actually gina-train ninyo inyong brain to sort information. Dili siya kanang murag mabutang gud mo og mga gamit sa inyong bag nga walay organizer. If you're going to organize your notes through algorithms and tabulations, class, you're actually training your brain to be able to unconsciously memorize the discussion. Okay? So here is a tabulation for the summary for the enzymatic glucose methodologies. Madami siya tingnan, class, pero actually, in general, tatlo lang talaga ito. Pwede siya glucose oxidase method, pwede siya hexokinase method, and pwede din siya glucose dehydrogenase method. Whereas itong dextrosticks and continuous glucose monitoring, class, is actually banking on these methodologies. Ang dextrosticks, it's enzyme impregnated, and typically, meron siyang glucose oxidase na enzyme. So ano yung principle for dextrosticks? It is reflectance photometry. For continuous glucose monitoring naman, class, is actually also similar to glucose oxidase. Pero rather than using the Trinder method that uses peroxidase, ang continuous glucose monitoring is electrochemical enzymatic sensing. So, ang enzymatic dyan, class, sa CGM is glucose oxidase. Ang electrochemical is with the use of, with the use of oxygen electrode. So, electrochemical, also polarographic ang CGM kasi it uses an electrode to measure the O2 concentration in your interstitial tissues. So now, let's focus on the other three major enzymatic glucose methodologies. Okay? So you have first your glucose oxidase. Ito yung reaction natin, class. You have two enzymes involved. Pero ito in particular is the Trinder method. Please do remember the term Trinder, class, kasi pabalik-balik siya. Typically kasi, when we refer to Trinder, it uses peroxidase and a chromogen. So, in Trinder method, class, what we measure is the oxidized chromogen. How do we measure? Through colorimetric detection. Again, class ha, typically, ang glucose oxidase, ito lang kasi yung glucose oxidase reaction. Now, what are we going to measure after? The coupled reaction after, class, could be peroxidase. So, if peroxidase yung gina-measure, you're going to have an oxidized chromogen, which you'll be measuring colorimetrically. Now, we also have the term polarographic assay. When we say polarographic, class, it's actually equal to saying electrochemical. Because what we are trying to measure here, class, is the O2 concentration. And ano nga yung O2 electrode? The O2 electrode, class, is what we refer to as the Clark electrode. Okay? Now, looking at the formula for glucose oxidase, asa na part nato ma-apply ang polarographic assay? Remember, class, that when we're going to use polarographic assay, ang meron 'di ba dito sa polarographic assay, class, is O2 consumption. So I think ito yung merong mga points for confusion. Consumption is not the same as concentration. Let's look at the reaction, class. Glucose plus oxygen plus water. By the action of glucose oxidase, there is the formation of gluconic acid and hydrogen peroxide. For a polarographic assay, class, we do not measure the product. What we measure is the loss of a substrate, which is oxygen. Okay? So, class, this basically means that if the concentration of oxygen is increased, then that means that O2 consumption is decreased. Nga no? Kasi if you're going to consume a lot of oxygen, then you would expect that concentration will decrease. Klaro ba, class?
Yes, sir.
Klaro. Sige. Now, unsa ang relationship sa oxygen sa glucose? Think of it like this, class. Ito. By the way, class, ang ganito na symbol, it means that concentration. So concentration of glucose and ito is consumption of O2. And ito naman is oxygen concentration. Okay? So using this representation, class, as the consumption of O2 increases, the concentration of glucose also increases. Or you could also say that as O2 concentration decreases, the consumption of O2 increases. Kasi nga 'di ba, class, if mas mataas ang weight, mas mababa sila on this example. Magets niyo ang analogy, class?
Yes, sir.
Alright, sige. Now let's move on to the hexokinase method. The hexokinase method, class, is a reference method. Bakit? Because of the specificity. Now, if itanong sa exam, class, ano ba yung nagbibigay ng specificity sa hexokinase method, what would you answer? It's actually not the hexokinase that gives specificity. Bakit? Let's break down the name. Hexo and kinase. Hexose. Remember, class, na hindi lang glucose ang merong C6H12O6 na chemical formula. Other hexoses, class, is actually capable of reacting with hexokinase. So, ano ba ang nagbibigay ng specificity sa hexokinase method? It's not hexokinase, rather it's G6PD. G6PD is glucose 6-phosphate dehydrogenase. Kasi, class, G6PD is specific only for this substrate, which is glucose 6-phosphate. Now, ano ba yung gina-measure natin sa hexokinase hexokinase G6PD coupled enzymatic reaction. What we measure, class, is ang NAD pH. Okay? NAD pH ang ato ang gina-measure diri a class. Meaning to say, we measure it spectrophotometrically. And if you remembered in our live discussion, I always emphasize that NAD pH increases in absorbance at 340 nanometers, whereas ang NAD P decreases absorbance at 340 nanometers. Majority kasi, class, ng enzymatic methods, whether it is glucose, lipids, proteins, they measure kasi, class, either NAD P or NAD pH. Basta, class, if merong H, increase in absorbance at 340 nanometers. Whereas if walang H, decrease in absorbance at 340 nanometers. Okay? Now, an increase in NAD pH and a decrease in NAD P in hexokinase is directly proportional to the glucose concentration. Okay? So, paano natin siya i-visualize? This is how you should visualize it, class. As NAD pH increases in concentration, glucose also increases in concentration. Meaning to say, they are directly proportional. Whereas, as NAD decreases, NAD pH increases. So, coincidentally, as NAD decreases, NAD pH increases, NAD pH indicates that glucose is also of the same concentration. So, this is how you visualize it. Then lastly, class, you have your glucose dehydrogenase for the enzymatic glucose methodologies. So it is similar to glucose oxidase, similar din siya with hexokinase. Bakit siya similar with glucose oxidase? Kasi you could use mutarotase. Ano nga ang role ng mutarotase, class? To convert the L conformation into the dextrorotatory conformation. Kasi, class, the problem with glucose dehydrogenase is that it is specific for the right-handed conformation of glucose. So what you do is to convert all L-glucose into D-glucose for it to be able to participate in the reaction. Okay? Then, it may be measured via spectrophotometry or colorimetry, and pwede din siya electrochemically or amperometry. So if spectro, class, then you're going to measure a colored compound. If electrochemically, then you could measure using the Clark electrode. Then, class, it is also similar with hexokinase. So, bakit siya similar with hexokinase? Because you could also measure NAD pH. And because you're going to measure NAD pH, class, is it increase or decrease in absorbance at 340? Increase. Increase in absorbance at 340 nanometers. Okay? So again, this is a summary for the enzymatic glucose methodologies. You have three methods in general: glucose oxidase, hexokinase, and dextrosticks. Of which, glucose oxidase, meron siyang dalawang coupled methods. It could be the Trinder method using peroxidase, or it could be polarographic. And when we say polarographic, class, we use the oxygen electrode. When we say Trinder method, class, we use peroxide and the enzyme peroxidase, and then resulting in a colored chromogen that has been oxidized. For hexokinase method naman, class, it uses a coupled enzyme that gives it its specificity. Ano nga yung coupled enzyme dito, class? Yun yung G6PD, which is highly specific for the substrate, that is G6P. And it is also the reference method. You also have your glucose dehydrogenase. Sorry, lahi di ako na circle. You also have your glucose dehydrogenase method, of which similar siya sa glucose oxidase. Bakit? Because you could measure it using spectrocolorimetry, you could measure it amperometrically, pero primarily, glucose dehydrogenase measures the oxidation of glucose with NAD, FAD. Basta the point lang nito, class, is you're still going to measure the reduced form. So yung mga NADH, FADH, all of which increases in absorbance at 340 nanometers. Okay? Then ang dextrosticks, it uses reflectance photometry, enzyme impregnated with glucose oxidase, and then it is banking on Trinder reaction. Kasi change in pH ang ginabasa ng reflectance photometry. Whereas ang glucose monitoring, kay actually still glucose oxidase but particularly polarographic assay. Bakit? Because it measures oxygen concentration in the interstitial tissues. Questions for enzymatic glucose methodologies? If wala, let's move on to the chemical glucose methodologies. So again, this is how you study it. You could either tabulate or you could either create an algorithm. So ilan ba yung glucose methodologies natin? As I've mentioned, you could divide it into two. First is the redox reaction. Second is the condensation reaction. Dito muna tayo mag-focus sa condensation. Kasi for condensation reaction, how many methods ba yung meron? Isa lang. You have your Dubowski method. Ano ba yung reagent na ginagamit sa Dubowski method? Orthotoluidine blue. Pero despite its name class, ang orthotoluidine blue, the positive reaction is green. Okay? Now let's look at the redox reaction, which we could divide into two. One is the alkaline copper reduction, another is the alkaline ferric reduction. But first, let's look at the alkaline ferric reduction kasi siya lang isa. Okay? You have here your Hagedorn-Jensen method. And ang color class, ang positive reaction is yellow. Yellow ba ang positive color sa Hagedorn-Jensen? So, the positive color actually class is the loss of yellow. Bakit loss of yellow? If you're going to look at the principle, ferricyanide to ferrocyanide, ferricyanide class is a yellow compound. Ferrocyanide is a colorless compound. So, as glucose concentration increases, ferricyanide concentration decreases, and ferrocyanide concentration increases. Because of an increase in the concentration of a colorless substance, then the positive reaction is a decrease in yellow color, indicating that the principle required for Hagedorn-Jensen is inverse colorimetry. Okay? Now, let's look at alkaline copper reduction, which has four methods. The first is the Folin-Wu method, which uses phosphomolybdate as a reagent, of which the positive color is phosphomolybdenum blue. Same with Nelson-Sumogi, but instead of phosphomolybdate, we use arsenomolybdate, and the resulting color is arsenomolybdenum blue. For neocoproin, madali lang siya class because Folin-Wu and Nelson-Sumogi blue sila. Ang neocoproin, kay just like Hagedorn-Jensen, yellow ang basehan. Pero in this case class, we use direct colorimetry. Bakit siya direct colorimetry? Because we measure the intensity of the yellow color. Meaning to say, as the intensity of color increases in neocoproin, the concentration of glucose also increases. Then lastly, you have your Benedict's method, which is a modification of Folin-Wu. Okay? Of which meron siyang gradient result, blue being negative, green being trace, yellow being positive, orange being 2 pos, and brick red being strongly positive. And class, please also be mindful kung ano ba yung environment to which these reactions occur. All of this reaction class, majority of this reaction occur in the presence of heat. Other than that, please look into the environment of which ano ba yung pH conditions. For condensation, it is acidic. Acidic because it uses glacial acetic acid. Ito naman sa alkaline copper and alkaline ferric reduction, as its name suggests, alkaline, this occurs in a alkaline medium. Now, just have a little bit of review class kung ano ba yung bumibigay ng alkaline characteristic. Ano ba yung substrate that gives it its alkaline character? Okay? So again, this is the tabulation for the methods of chemical glucose determination. You have alkaline copper reduction, alkaline ferric reduction, and condensation methods, of which Folin-Wu and Nelson-Sumogi has blue color. Neocoproin is appearance of yellow. Hagedorn-Jensen is disappearance of yellow. Benedict's test is a gradient of color, of which red or brick red is the strong positive reaction. And orthotoluidine blue, which is the reagent for the Dubowski method, a type of condensation technique that occurs in acidic environment, the positive color is green. So here are the key differences among methodologies between enzymatic and chemical methods. Ang specificity for enzymatic methods class is mataas. Bakit? Because enzymes are glucose-specific, especially hexokinase G6PD coupled enzymatic reaction. So kayo na magbasa ng ibang differences nila. Question so far for carbohydrates class. Are there any clarifications?
Wala na, sir.
Okay. So for the last part of our discussion class, atoan na siyang idiretso for lipids, okay? And according to the survey, majority of you also found it difficult in terms of the methodologies. Okay? You have your cholesterol methodologies and triglyceride methodologies. So sa lipoprotein, wala man kayo nahirapan. Siguro alam niyo na yun kasi na-discuss naman siya sa lecture. So this is the triglyceride methodologies. For chemical methods class, you have the Van Handel and Silversmith as well as the CDC method. Now, to avoid confusion class, ganito siya. Van Handel and Silversmith is the old method. CDC method class is the same as modified Van Handel and Silversmith. Okay? Ang modified Van Handel and Silversmith is the same as the CDC method. So class, ito siya, itong nakalagay dito na modified CDC, actually dapat wala na yung modified because in the first place, modified Van Handel Silversmith is the CDC method. Klaro ba class?
Yes, sir.
Alright. Sige. So, if we're going to use the Van Handel Silversmith reaction, you're going to have blue as a positive color. But if you're going to use the modified Van Handel Silversmith or the CDC method, you will have a pink positive reaction. Either way, they are measured in 570 nanometers. And Van Handel Silversmith is the reference chemical method. Okay? Another thing, class, is that the Van Handel and Silversmith also has been modified para hindi spectral wavelengths siya babasahin. You could also measure using fluorometric techniques. Paano? The resulting formaldehyde is made to react with diacetyl dihydrolitidine para magkaroon ng yellow fluorescence, which is measured fluorometrically. Increase in fluorescence also indicates increase in the concentration of triglycerides. Between spectro and fluoro kasi class, the fluorometry also increases the sensitivity. Okay? So that is for the chemical methods. Now let's look at the enzymatic methods. For enzymatic class, ito talaga yung general. Wait lang. So you have two methods. You have glycerokinase plus pyruvate kinase and lactate dehydrogenase. You also have glycerokinase plus glycerophosphate and the diaphorase method. So before we discuss this table, I think it would be easier if dito na lang tayo sa algorithm titingin. Kasi class, di ba kanina you have there glycerokinase method in general. The in general glycerokinase method class, ito talaga yung reaction na nangyayari. Triglyceride is broken down by lipase to form glycerol and fatty acids. Between glycerol and fatty acids class, ano nga yung hindi typically nag-exist in the blood? Glycerol ang typically class hindi nag-exist freely in the blood. Typically class ha, take note of the word typically. Meaning to say meron siya but not in the same way as fatty acids. Kasi fatty acids class occur in the body in the form of FFAs, free fatty acids. The reason why we opt for glycerol kasi in one molecule of triglyceride, you only have one glycerol molecule and 3 molecules of fatty acids. Okay? So ang glycerol class, once glycerol is liberated, it is phosphorylated. It is phosphorylated by glycerokinase by taking one phosphate group from ATP. The result is glycerophosphate and adenosine diphosphate. Now, from this end reaction by glycerokinase, you could opt for two methods. One is through the pyruvate kinase lactate dehydrogenase method, and the other is through the glycerophosphate dehydrogenase and diaphorase method. Let's first look at the more easy reaction, pyruvate kinase and lactate dehydrogenase. ADP is made to react with phosphoenol pyruvate by the action of pyruvate kinase. So, from its name, pyruvate kinase class, it's going to move a phosphate group from pyruvate from phosphoenol pyruvate, ibalik niya sa ADP. The resulting is adenosine triphosphate and pyruvate, of which the dephosphorylated pyruvate is now made to react with NADH. And the resulting end product class is NAD. Ano nga ang wavelength na gina-measure ang NADH and NAD class?
340 nanometers, sir.
340 nanometers. Now, look at the reaction. NADH is a substrate. NAD is a product. And NAD ang gina-measure natin. Is it increase in absorbance at 340 or decrease in absorbance at 340?
Decrease sila.
Decrease in absorbance. Meaning to say, class, as the concentration of NAD increases, the absorbance at 340 nanometers decreases. What does that tell you about the concentration of triglyceride? Is the concentration increase or decrease, class? Again, ang scenario is mataas ang NAD, bumaba ang absorbance. What does that tell you about triglycerides? Is it increase or decrease? This would increase. Okay? So, let's try to put that into perspective. Nganong mag-increase ang triglycerides, class? Think of it this way. Okay. Ingani ni siya, class. Class, if madami ang raw materials nyo, madami din ba ang products na nagagawa nyo?
Yes, sir.
Yes, sir.
Alright. In this case, triglyceride is a raw material. Triglyceride is a substrate. So, madami kang pwedeng gawin na product. In this case, the product is NAD. Meaning to say, class, as NAD increases in concentration, it is attributable to the fact that triglyceride is increased. Meaning to say, as the concentration of NAD increases, then you could also say that triglyceride is increased. The problem, class, is that ang NAD kasi, class, it has decrease in absorbance at 340 nanometers. Meaning to say, class, if absorbance is increased, if absorbance is increased, that means, class, that NAD is decreased. But, like I said, as NAD concentration increases, absorbance is actually decreasing. Magets nyo, class? Kasi di ba if mas maliit ang value ng absorbance, lighter siya. That is why nakaangat siya. Then ang NAD class is nag-increase daw ang concentration. As the concentration of NAD increases, nagbababa ang absorbance. Kaya, class, on this analogy, mas mabigat ang triglycerides in NAD and nagbababa ang value. Magets niyo, class? Kasi we will now be moving on to the glycerophosphate dehydrogenase and diaphorase. Now, for glycerophosphate dehydrogenase and diaphorase class, still it is a two reaction. What happens? If sa pyruvate kinase lactate dehydrogenase is ADP yung substrate from the previous reaction, sa glycerophosphate dehydrogenase and diaphorase class is ang glycerophosphate ang substrate sa next na reaction. Kaya nga, class, glycerophosphate is made to react with nicotinamide adenine dinucleotide by the action of glycerophosphate dehydrogenase, resulting in the formation of dihydroxyacetone phosphate, NADH, and H. Okay, so you might be wondering nga na broken lines ang NADH dira. Later na nako na siya i-explain. So, with the production of NADH, NADH is then made to react with tetrazolium di. And by the action of diaphorase oxidase, formazan is produced. Now, ang end product ng reaction for glycerophosphate dehydrogenase diaphorase class is formazan. Okay? Ang formazan class, it is a colored chromogen. Colored siya. So you're going to measure that colorimetrically or spectrophotometrically. Pero typically, class, colorimetry ni siya. Kasi when we say spectro kasi, class, dapat spectral ang wavelength. Typically, ultraviolet ang wavelength. Now, why ngayon ako gibutangan og broken line ang NADH? you could actually end the reaction here. Pwedeng wala na to. Pwede na ni siya wala. Ngano? Because we could measure NADH. Ano nga yung wavelength that we measure NADH, class?
340, sir.
340. Is it decrease in absorbance or increase in absorbance?
Increase.
Increase in absorbance. Knowing, class, na NADH is increase in absorbance at 340, as NADH concentration increases, what happens to the absorbance at 340? Increase or decrease?
Mag-increase, sir.
Mag-increase. Now, what does that tell you about the concentration of triglycerides?
Mag-increase po, sir.
Mag-increase. Okay. Ngano mag-increase? Because in this case, class, NADH ang gina-measure natin. NADH is the byproduct. Dito kasi, class, NAD ang byproduct. Makita nyo ang difference? Both of them are measured at 340 nanometers. Pero ang difference, class, is yung absorbance nila sa 340 nanometers. NADH has an increased absorbance at 340. NAD has a decreased absorbance at 340. But both of them, the concentration of NAD, ganito, class, ha, if product sila, both kasi sila product. And remember, if the substrate is increased, then you would expect that the product is also increased. Meaning to say, class, an increase in the concentration of product, whether NAD or NADH, indicates that the substrate is also increased in concentration. Ang interpretation, class, is nakadepende na lang siya sa absorbance. NAD8, ang NADH, class, ito, tataas yung absorbance niya 340. Ang NAD, bababa ang absorbance niya 340. But it does not change the fact that the concentration of NAD is increased. Klaro ba, class?
Yes, sir.
So it really is just banking on your interpretation of the absorbance. Okay? Yung for glycerophosphate dehydrogenase as well as the diaphorase reaction, I want to emphasize that typically if you're going to use the diaphorase oxidase, the endpoint is formazan, which is measured colorimetrically. But as I've mentioned, you could end the reaction here and simply just read NADH. Read NADH at 340 nanometers, of which NADH has an increasing concentration at 340, as opposed to NAD na decrease ang absorbance at 340. Okay? Again, so this is the summary for the triglyceride methodologies. You have your chemical methods, enzymatic methods. Ang enzymatic typically uses lipase and glycerokinase. And then, ang glycerokinase, it could be coupled to other enzymes such as the PKLDH as well as the GPO and diaphorase method, of which we measure NAD at 340 nanometers, of which as NAD concentration increases, there is a subsequent decrease in absorbance at 340 nanometers. For the GPO diaphorase method, the end product is formazan dye, which is measured photometrically, colorimetrically at 500 to 600 nanometers, of which, as I've mentioned, you could also end the reaction just by measuring the NADH at 340 nanometers, which causes an increase in absorbance. For the reference methods, class, if ang tanong, class, is ano yung reference method, you would answer GCMS or IDMS. Pero if ang tanong is ano yung reference chemical method, you would answer the Van Handel and Silversmith. Questions so far for triglyceride methodologies? Mas nakaintindi ba, class, sa NAD and NADH na measuring?
Yes po, sir.
Yes, sir.
So now let's move on to cholesterol methodologies, of which we have a variety. You have your Lieberman-Burchard, your Salkowski, Abel-Kendall, your enzymatic methods, your electrochemical methods, and the reference methods. Okay? So dito muna tayo sa Lieberman-Burchard and Salkowski. Actually, there's nothing much I could discuss here because this is raw memorization. So at least be familiar with the substrate that is being used, and especially dakong sala, class, if malimtan ninyo ang color reaction. Lieberman-Burchard, green, Salkowski is red. Okay? What we want to focus on is the Abel-Kendall, which I want you to be able to memorize. Dakong sala, if dili niyo ma-memorize, ang steps for Abel-Kendall. Okay? So this is the modified Abel-Kendall, class, which is a 3-step process: saponification, extraction, and colorimetry. Ito siya, class, this is an enzymatic reaction using cholesterol oxidase. So, this is still, class, the same principle lang naman sa sa modified Abel-Kendall, saponification, extraction, and colorimetry. Actually, the same principle ang ginagamit din natin sa enzymatic methods, but we increase the specificity by including enzymes. So I apologize for the confusion. Balik muna tayo dito sa Abel-Kendall. Kasi 'di ba, we use a saponifying agent, we use an extracting agent, and we also use a then we measure using colorimetry. If you remember class, ang principle ng saponification is the liberation of cholesterol from its storage form in the body, which is cholesterol esters. Now, let me compare that. Remember class that in saponification step for the modified Abel-Kendall, ang goal ng saponification is to liberate cholesterol from cholesterol ester. The same in the enzymatic reaction. But rather than using a chemical, what we are using is cholesterol esterase. So again, saponification talks about the liberation of cholesterol from cholesterol ester. For extraction naman class, for the modified Abel-Kendall, di ba ang ginagamit natin dyan is N-hexane. N-hexane. Yung N-hexane na yan class is basically just to liberate yung cholesterol and extract non-cholesterol. I-remove ang non-cholesterol. Kaya para ang cholesterol lang ang mag-react during colorimetry in the modified Abel-Kendall. Sa extraction step here in the enzymatic assay class, it's not something that we have to do. Bakit hindi na kailangan ng extraction step sa enzymatic method? Because the enzymes are highly specific. It will not react with non-cholesterol substances. And for the enzymatic method class, you have your cholesterol esterase and cholesterol oxidase. And for the last part, it is banking on the Trinder reaction. Bakit siya Trinder reaction? Because with the help of peroxidase, we could actually oxidize the 4-amino antipyrine in the presence of phenol to form the quinonimine dye, which is oxidized. Okay? So ito siya class, itong slide na ito, this is just to emphasize na there is similarity in the chemical and enzymatic methods. Ang difference lang class is kung ano yung reagents na ginamit. For saponification ng modified Abel-Kendall, chemical lang siya, kung ano yung nasa PowerPoint. For the enzymatic methods naman class is rather than using a saponifying chemical, what we use is an enzyme cholesterol esterase. Ano nga ang role ng saponification? To remove cholesterol from cholesterol ester. Okay? For extraction naman, we use N-hexane in the chemical method. Then for enzymatic method, there is no need for extraction because in the first place, the enzymes are highly specific. And then both of them, modified Abel-Kendall and the enzymatic method, is measured using colorimetry. Pero class, there is a modification. There is a modification in another enzymatic method. Okay? So mali ni siya, dili ni siya modified Abel-Kendall. So in this slide class, we want, this is still an enzymatic assay, pero rather than measuring the end product quinonimine dye, we could just measure O2, oxygen concentration. So, yes, there is saponification by the addition of cholesterol esterase. And we end the reaction here because we just measure O2. So, it is a polarographic assay. Ano nga yung electrode na ginagamit natin for the measurement of O2 class?
Clark, sir.
We use the Clark electrode. Now, class, is O2 a substrate or a product?
Substrate, sir.
In this case, O2 is a substrate. Tama. So, and then class, if you're going to look at the reaction by cholesterol oxidase, you're going to use up O2. Meaning to say, as O2 concentration decreases, what happens to the substrate cholesterol class? Is it increase or decrease?
Increase, sir.
Increase ang cholesterol. Ngano increase ang cholesterol? Because if decreased ang O2 concentration, then that's equal to saying increased ang O2 consumption. So, as O2 consumption increases class, the O2 concentration decreases, which tells you that cholesterol is increased. The substrate cholesterol is increased. Okay? Question so far? Wala na. Okay. So again, ito yung mga methodologies natin, Lieberman, Burchard, Sarkowski, Abel, Kendall. Then our reference method class is GCMS, IDMS. The same lang ang cholesterol and triglyceride ng reference method. But, ito class ha, to clarify, reference method in general is GCMS, IDMS. But if ang tanong class is what is the reference chemical method, for triglyceride, you will answer the Van Handel-Silversmith. For cholesterol, you would answer Abel Kendall. And please class, do not forget the term modified. Modified Abel Kendall, modified Van Handel-Silversmith, or the CDC method. Questions for cholesterol?
Wala,
None?
sir.
Okay. So let's have our question and answer portion, class. Meron ba kayong mga tanong sa other parts of the discussion na wala natin na-cover sa ating review? Clarifications for other areas na medyo blurry pa, class. So I'm assuming na wala. If not, let's check at our direction for finals. So I've asked you, class, do you think that you can better study if the pre-lab discussion for the finals term is pre-recorded? And according to 45 responses, 77.8% of you would agree. And actually, class, I would also agree na mas makastudy kayo. Kasi you have the liberty of time kung kailan kayo mag-aaral. And additionally, and class, naka-record naman po good ko. Naka-record na ako ng pre-lab discussion for proteins, for renal function. Okay? Then, ang kulang na lang, class, for finals is liver function and trace elements. Dalawa na lang ang kulang, and actually I could finish liver function siguro tomorrow or tonight if wala akong gawin. And ang trace elements, I think last topic naman siya for final, so I have taas-taas pang oras ko to make that. So basically, class, even karong midterms pa lang, you could actually already watch the proteins and renal function. Kamu na bahala kanus ninyo siya tanawon. Basta I've already sent class yung schedules natin for the assessments.